All right, so light-independent reactions. This is the second half of photosynthesis. We already talked about the light reactions. These are the ones that don't need light to happen. All right, so it is called, like another name for it, is the Calvin cycle. So there's this dude, Calvin. He's a really old scientist. He discovered this thing that's happening inside of the chloroplast. As scientists do, they are generally pretty vain people. I discovered this thing, I'm going to name it after myself. So that's why it's called the Calvin cycle. The reactants and products of this, so CO2, atmospheric carbon dioxide, is going to go in, along with that ATP and NADPH we produce in our light reaction. And those three things are going to interact and react with each other to produce sugar or glucose, ADP, and NADP+. Remember that ADP and NADP+ are going to turn into reactants for our light reaction. So the very main reason, sorry, this thing is bothering me. The very main purpose of this is to build sugar. All right, sugar means bonds can be broken, energy can come from that. When we're making this sugar, we need to use energy. And that energy is coming from our ATP that we just made in our light reaction, and the NADPH, which holds electrons, which are another type of energy. So our electron carrier is carrying those electrons from the light reaction over to our Calvin cycle. So there are three steps: carbon fixation, reduction, and regeneration of that starting molecule. So it's a cycle. That means things are going to keep going and going and going. We're not going to get rid of all of our stuff. We're going to reuse some of it to start a new cycle again. So carbon fixation, our very first step here. We're going to have to do something called fixing carbon. Atmospheric carbon dioxide, we can't use. Plants can't just use it as it is. They have to do something called fixing it. They have to break it down into a molecule that they can use, an organic molecule. So in this process, they're going to fix that carbon in a way that the plant can use it later. This process, again, needs energy. So we're going to be using some ATP and NADPH that we've made in the light reactions. The Calvin cycle is taking place in the stroma. So remember, the stroma is that place outside the grana. The grana are those stacks of thylakoids, but it's still within the chloroplast. So it's dark. There's no light getting in there. But that's okay, we don't need light. This is light-independent reactions. So let's look a little bit closer here. Let me get my pointer. All right, so a CO2 molecule up here, a CO2 molecule is going to combine with a 5-carbon molecule called RUBP. So over here, we have RUBP. It's coming into contact with a new CO2 molecule coming in from the atmosphere. And they're going to be catalyzed. Remember, enzymes are catalysts. They're going to be our workers. They're going to put those molecules together. And that molecule is called rubisco. That enzyme is called rubisco. And enzymes are often named for the things they do. So rubisco, kind of matches with this RUBP molecule here. So those you kind of can figure out they go together because they have very similar names, rubisco and RUBP. This is going to happen 3 times because we're bringing in 3 carbon dioxide molecules. So for every one of those carbon dioxide molecules, we're joining it with one of these 3 RUBPs. So 3 carbon dioxides, 3 RUBPs means this carbon fixation happens 3 times. The result, so we have this 5 carbon RUBP, 1, 2, 3, 4, 5 carbons. That is combining with 1 more carbon from that carbon dioxide. We're left with 6. So we're trying to count these carbons, make sure everything that matter is conserved. So after the rubisco pushes these 2 things together, we get this new 6 carbon molecule. But the rubisco does one more thing before it lets it go. It now breaks it in half. I know, it just put it together, but it's breaking it into half so that we have 2 3-carbon molecules. Each of those carbon molecules has a phosphate group, so it's just cutting it right down the middle here. So we get 2 3-carbon molecules with a phosphate group from one of those 6 carbon molecules that was made. Lots of numbers, lots of letters, I know, a little bit more. So since we have 3 carbon dioxides merging with 3 RUBP molecules, we're going to get 3 of these 6 carbon compounds. And those 3 6-carbon compounds are broken in half. So now we have 6 3-carbon molecules. Lots of counting, I know. And these 3 carbon molecules are called 3, for their 3 carbons, PGA. Again, you don't need to know what PGA stands for or RUBP stands for. They're long, like 3 phosphoglyceric acid. Like even I have trouble saying it. Just know that the letters stand for something, but you don't need to know what those letters stand for. All right, so in summary, I know there's a lot of stuff. I'm just going to keep repeating it and repeating it to sort of help you get it in your brain here. So 3 carbon dioxides, remember we're doing 3, enter the chloroplast. Then we're taking 3 of these RUBPs and a Rubisco enzyme, mushing them together to create a 6-carbon molecule. And then before it lets go, that Rubisco is going to break it in half to form 2 3-PGA molecules. 3 for those 3 carbons. PGA was that really long name for that molecule. So we have 6 of those 3 PGAs because we're breaking that 6-carbon in half. Okay, next step, reduction. We're going to be reducing something. So in this second stage, this is where we're going to use our ATP and NADPH that we just made in our light reaction. They're going to be used, their energy is going to be used to convert those 3 PGAs we just made into a different molecule called G3P. We're not actually creating something, we're just rearranging some atoms in that molecule. But in order to rearrange that stuff, we're going to need some energy. That NADPH, remember it's carrying some electrons. We're going to break off that hydrogen to release those electrons. And when we do that, that's called reducing that NADPH. So that's why we get the reduction name here. So here's a different look. We have some energy going on here. So we have our 6 molecules of 3-PGA. I know there's only 2 shown here, but there are 6 of them. And here is the energy we're using. So there are 6 of these molecules, so we need 6 ATP. They're going to be broken apart, that one of those phosphate groups is going to be broken to get energy to do that. And we'll be left with 6 ADPs. And we're also going to use some of those electrons from NADPH. Again, that H is going to break off, release those electrons, and we'll be left with NADP+. This is the energy we're using to rearrange our 3 PGAs. The result is something called a G3P molecule. Again, 3 because we have 3 of those carbons, P for that phosphate group. And we're going to have 6 of these. Remember, we started with 6 of these PGAs, so we're going to be left with 6 of these G3Ps. One of those 6 is going to leave the cycle and become part of a glucose. But we're left over with 5 of these G3Ps. Well, what is their job now? We're going to find out. So in summary, this reduction, we have our 6 molecules of 3-PGA, and we're going to use ATP and NADPH, 6 each of those because we have 6 each molecules of 3-PGA, and we're going to rearrange the PGA into G3P. One of those G3Ps is leaving the cycle to become glucose. The 5 that are remaining are moving on to the next step. All right, this last step called regeneration. So in our cycle, we're going to have to regenerate some stuff so we can start the cycle all over again. Here's where it's going to happen. So now we're left with our 5 G3Ps because one of them left to become glucose. That's not useful for this cycle. But now we're left with 15 carbons. So we have 5 of these 3 carbons 5 times 3 is 15. So we have 15 3-carbon molecules left over. We want to transform those into our RUBP so we can start all over again. And if we're transforming and moving molecules around, we need energy. So we're going to create 3 RUBPs. So we need to use 3 ATPs. We're left with 3 RUBPs because, again, conservation of matter. We have 15 carbons total. 5 3-carbon molecules called G3P. We're rearranging them into 3 5-carbon RUBPs. Lots of numbers, lots of letters. It's really confusing. All of these carbon shuffles around. Things are leaving, things are attaching, things are shuffling. So often I've heard it called the carbohydrate scramble. So we're trying to make a carbohydrate, our glucose, and in order to do that, we got to scramble some stuff around, all these carbons messing around in this Calvin cycle. So I've heard it called the carbohydrate scramble, which I think is kind of cute. But this cycle is crazy. Lots of letters, again, lots of numbers. So in summary, our regeneration, our last step in this cycle, those 5 leftover G3Ps are going to be rearranged into 3 molecules of RUBP. In order to rearrange these, we have to use ATP. So one cycle of the Calvin cycle, we get 1 of those 3 carbon molecules leaving to make glucose, right? Well, glucose has 6 carbons. So we need 6 turns of this cycle to get 1 molecule of glucose. Yeah, doesn't feel particularly efficient, but it's what works. So if it ain't broke, don't fix it. So here's the overview. Again, we have 3 carbon dioxide from atmosphere coming in, bonding with that RUBP in the Revsco enzyme. Then that Revsco is also going to break it in half, giving us our 3 PGAs. We're going to use ATP and NADPH as energy to rearrange those PGA molecules into G3Ps. 1 of those G3Ps is leaving to make glucose. 5 of those G3Ps are being recycled back into the Calvin cycle. We need to rearrange them so we can turn them back into RUBPs. To do that, we need to use energy or ATP. Whew, that was a lot. All right. I'm going to stop my recording.